The cell cycle is a series of events that takes place in a cell as it grows and divides. A cell, it spends most of its time in what is called an interphase. And during the times it grows, it replaces the chromosomes and prepares for the cell division. Then the cell then leaves interphase and undergoes mitosis and completes its division. I'm going to talk to you how the cell cycle actually does exist in the world and what its purpose. The cell cycle consists of 4 stages: gap 1, synthesis, gap 2, and mitosis. The first one we go over is gap 1 consists of a growth and copying of organelles, and it makes molecules building blocks. It's a first of the 4 phases in the cell cycle that takes place in the irregular cell division. In this part in interphases, the cell synthesizes in preparation for the central steps leading to mitosis. Synthesis, it copies DNA and new controls the symbols. It's the second phase of the cell cycle, which means the DNA is replicating between the requirements of G1 phase and the G2 phase. Since it was accurate that duplications of the genoid is crucial to the eventual successful cell division. The process of that occurs during the S phase is tightly regulated and widely conservative. Gap 2 is the additional growth of the molecule. It's essentially the last part of the interphase. The cell has grown and the DNA has been replicated, and now the cell is almost ready to divide. This is the last stage, and the reason why it's the last stage is because it's all about its preparation for the mitosis of the meiosis. During the G2, the cell has time to grow some more and produce any molecules that still needs to divide. Mitosis is the part of the cell cycle which the replicated chromosomes are not separated to the nucleoids. The cell division, it gives the rise to genetic identical cells in which the total number of chromosomes are maintained. They have 4 other stages within the separate stage. The first stage is a prophase. Within the stage, chromatin it considers into chromosomes with certain spindle fibers form. Metaphase is where the chromosomes line up along the middle. They're connected by the record tubes and correctly attached to the center. Anaphase is a sister chromatid where each chromosome are separated opposite to the size of the cell. Telophase is the splinter fiber or dissolve in a nuclear envelope of both sides. The nucleoids reappear in chromosomes uncloyed. During the telophase, the cell plate forms across the cell end in the area where the old plate from the metaphase stage. Cytokinesis is a part of the cell division process which the cytoplasm from a single irregular cell divides into 2 daughter cells. Cytoplasm division begins during and after the late stage of the nuclear division of mitosis and meiosis. It's considered to be a separate part from the mitosis. It's a separate part from the cytoplasm. It's a physical process of the cell division. It contrasts ring strings and pitching the plasma membrane, forming a caudal ferrule. The G1 checkpoint is where the cell decides on whether it's on a process the division based on the cell size and nutrients. This is where the urocoids typically arrest the cell cycle if environmental conditions make the cell division impossible, or if the cell passes on to G0 for an extended period of time. Such things as the animal cell, the checkpoint is what we call a restriction point, and where the yeast cells is its start point. The G2 checkpoint ensures that all chromosomes have been replicated and that the DNA is not damaged before the cell enters the stage of mitosis. It prevents the cells from entering the mitosis when the DNA is damaged. It provides an opportunity for repair and stopping the peripheral of damaged cells. Because of that, it helps to maintain its genomic stability. It's an important focus that understand that the molecules causes cancer. So it really helps for like the damaged cells or the crazy like cell birth. The M checkpoint occurs near the anaphase stages of the mitosis, and it is also known as the spindle checkpoint because it determines whether the sister chromatids are correctly attached from the spindle. That is all for the cell cycle.